Thanks for tuning in. In this video, I'll discuss the results of our recent preprint that showed promising effects of sunflower on honeybee resistance to Varroa destructor mites, one of the most maligned pests of managed bees worldwide. For the last 40 years, beekeepers around the world have been plagued by the mite Varroa destructor. Originally a parasite of the Asian honeybee Apis cerana, these mites have now spread through managed Apis mellifera worldwide. The mites reproduce in honeybee brood cells and feed on the fat body of pupae and adults. They transmit deadly viruses and are strong drivers of colony losses. Existing control methods include breeding of bees for mite grooming behavior and excavation of infested brood before mites can reproduce, fumigation of colonies with arachnid targeting compounds, and ensuring that bees have adequate nutrition to cope with the viruses that mites transmit and compensate for the physical drain of having their innards constantly fed upon. These methods keep mites in check, but the frequent application of these acaricidal compounds is expensive, labor-intensive, and can have off-target effects on bees themselves. As a result, development of new mite control strategies is an area of active research. Allowing bees to self-medicate against parasites by supplying them with medicinal plants could offer simultaneous nutritional and antiparasitic benefits, and might circumvent the labor costs and possibly some of the side effects associated with application of miticides. Sunflower pollen could provide such a resource. This plant is native to North America, but cultivated as an oilseed worldwide. Bees that specialize on pollen of plants in the sunflower family, Asteraceae, have been noted for their resistance to brood parasites. Sunflower pollen recently showed promising effects against gut infections in bumblebees, with sunflower cropland negatively correlated with infection intensity of bumblebees in Massachusetts. In honeybees, there is some evidence for self-medication with sunflower honey when bees are infected with nosema. Here we use two types of experiments to investigate the relationship between sunflower availability and Varroa mites in honeybee colonies. First, we evaluated correlations between the amount of sunflower cropland surrounding apiaries and the levels of Varroa infestation during and immediately following the sunflower bloom period. To do this, we combined land cover data with mite counts from the National Honeybee Health Survey. Then, to test for a causative relationship between sunflower pollen and mite resistance, we experimentally fed colonies with either sunflower pollen, wildflower pollen, or a pollen-free protein supplement. The landscape survey showed a negative correlation between sunflower crop area near apiaries and mite counts, with a 28% decrease in mite counts for every doubling of sunflower cropland within 2 miles of apiaries. In the experimentally supplemented colonies, the sunflower-fed colonies had 2.8-fold lower mite counts after 5 weeks relative to the protein supplement-fed controls, and none of the sunflower-fed colonies exceeded the 3 mites per 100 bee treatment threshold. This suggests a causal role of sunflower in mite resistance. So these results indicate that sunflower crops and their pollen could provide a key nutritional resource for mite-infested honeybees. They also suggest further investigation into how antiparasitic plants, which have been shown to provide transgenerational protection against predators and parasitoids in other insects, such as monarch butterflies, could enhance Varroa resistance in honeybees. Deliberately planting such resources could offer an environmentally friendly way to mitigate colony loss, while also providing food for other pollinators. However, sunflower cropland in the US has been declining by 2% per year since 1980 and 3.5% per year since 1995, which is likely reducing honeybee access to this potentially beneficial resource. This decline reflects changes in land use in the upper Midwest, which is where most of our nation's sunflowers are grown. Much of the agricultural area that was previously devoted to bee-friendly crops, such as sunflower and canola or brassica, has been converted to corn and soybeans for production of biofuels and animal feed. Land use policies that reverse this decline could benefit mite-plagued managed honeybees as well as Asteraceae specialists. Given the promising results here, follow-up studies are warranted to investigate the mechanisms underlying the effects of sunflower on mites seen here. Identifying the mode of action could allow development of sunflower-based or sunflower-inspired colony supplements. These could offer the benefits of sunflower pollen to bees in seasons and regions where sunflowers are unavailable. One possibility for a mode of action relates to the unusual sterile composition of sunflower pollen, which includes some compounds that are toxic to other insects. Honeybees are unable to synthesize their own sterols. Instead, they incorporate sterols from pollen into their tissues. The mite sterols reflect those found in the pupae that the mites feed upon. So if mites are more susceptible to sunflower sterols than honeybees are, feeding bees with these sterols could be a way to selectively target Varroa mites. On the other hand, sunflower has a reputation for being a low quality and a low protein food source for most generalist bees, including honeybees. It could be that the brood of these sunflower-fed colonies are lower in nutritional quality for mites, but also not very healthy or numerous overall. In this case, consumption of sunflower pollen, just like any medicine with side effects, would have both costs and benefits. But the benefits could outweigh the costs when mite pressure is high. We need more information on how sunflowers' effects against mites are achieved and at what price in order to better develop dietary supplement or landscape-based interventions to enhance resistance to these pests. Nevertheless, our results here suggest that reversing declines in sunflower cropland could benefit not only bees that specialize on sunflower, but managed honeybee colonies as well. That means that antiparasitic plants could offer an alternative or a complement to existing methods of mite control. Now for your poetic summary. Fields of sunflower blooming in sight yield for many a bee a delight, but with bright yellow joy displaced by corn and soy, honeybees could lose balm for their mites. I'd like to thank my numerous co-authors, including Lynn Adler, who coordinated this full study, the Bee Informed Partnership for providing data from the Honeybee Health Survey, and the USDA NIFA for funding. Thanks for watching. I look forward to seeing your comments in the chat.